Megan's sending a nice little message to us as we get started here. Hello, everyone. Welcome to the Collaborative Science for Estuaries webinar series. This is Nick Sobral with the Science Collaborative, and I'm joined remotely by Megan Brass, who's also running support for the webinar for today. Thank you for joining us for this month's webinar, which is titled Understanding the Interconnectedness of Climate Change, Salt Marsh Resilience, and Nuisance Mosquitoes. Some quick introduction on the reserve system. The National Estuarine Research Reserve System is a national network of unique research reserves as shown on this map. This is a NOAA program that works in collaboration with a local place-based partner, either a state agency, university, or nonprofit. Each reserve site includes programs focused on land stewardship, research and scientific monitoring, training programs for the public and local officials, and education. The Science Collaborative supports science for estuarine and coastal decision makers by coordinating regular funding opportunities and supporting end-user driven collaborative research, assessment, and transfer activities. For today's webinar, all attendees were muted upon entry and will be handling questions via the question feature in the GoToWebinar console. We'll have time to answer questions at the end of today's webinar, but we encourage you to submit them as they occur to you throughout the webinar by just entering them into that little field on the questions box there on the console. So before we get started, just a quick snapshot of the registration as it stood yesterday. I checked yesterday afternoon. I checked again this morning as well, but these charts are based off of yesterday's numbers. And we're really happy to see so much interest from across all over the country, really, but especially in just the East Coast was a huge standout to me. So it's great to see so much interest and from a range of organization types and regions, not just within the Mid-Atlantic region where the research took place, but also outside of it. And as always, a big welcome back to returning attendees and thank you for joining us to all newcomers. So with that, I will go ahead and introduce our speakers for today. Today's webinar will include presentations from these 4 team members who will go over the different aspects of this project. After that, we'll end with some Q&A with the speakers. So joining us for today's webinar, we have Richard Lathrop, Lisa Auermuller, Kaitlin Gannon, and Dina Fonseca. Rick is a professor of ecology and watershed monitoring, whose research focuses largely on water resources and providing coastal communities with science and tools for decision-making in the face of climate change and sea level rise. Rick served as the project lead for this project, coordinating the many aspects of the project and leading the mapping and modeling elements. Dina is a molecular medical and veterinary entomologist. She primarily develops tools to reveal incipient infestations, identify which traits are associated with expansion and damage of invasive species, and optimize management strategies. In this project, Dina served as the project co-lead, coordinating the development of the mosquito eDNA assays. Lisa served as the collaborative lead for the project and is the assistant manager at the Jacques Cousteau Reserve in New Jersey, where her duties include assessing the needs of coastal decision makers and providing relevant and timely training opportunities. And last but certainly not least, Kaitlin is the education coordinator for the Jacques Cousteau Reserve. She has extensive training in wildlife interpretation and conservation, and she supported outreach efforts for the project and developed the educational module for use in teachers on the estuary or TOTE training. And with that, I will go ahead and pass it to our first speaker, who I believe is Lisa, and look at her all ready to go. Awesome. We give you keyboard and mouse control, Lisa. And the floor is yours.
Okay. Thank you so much, Nick. It's great to be with so many of you today and many names that I recognize, which is always nice to see. Hopefully we'll get to see each other in person sometime soon. I'm gonna set the stage for our webinar today. Here's a picture of a gorgeous New Jersey salt marsh. This happens to be the Sentinel site area of the Jacques Cousteau Reserve in Tuckerton, New Jersey. And if you were to look at that same salt marsh from a different perspective, you would see how the marsh looks a little different from up in the sky. For those of you that are familiar with salt marsh processes over time, they're certainly very culturally important, being areas in New Jersey where they were salt hay farmed for many years. We have many lagoon communities built into our salt marshes, at least here in New Jersey, probably a little bit of a slightly different lagoon than those of you in Florida are used to hearing about, but this is what a lagoon community in New Jersey looks like. And then something that's very important to the webinar topic today is the mosquito management activity that has been done on our salt marshes here in New Jersey to protect the public health of the public within the coast. And it is some of those uses of the salt marsh and our desire to protect them and restore them, that really is the impetus and the thought behind why we originally applied for and got this project that you're going to hear about today. So as I said, we took a look as a team at the salt marsh structure holistically. We then thought about all the different salt marsh mosquito management activities that have happened on the marshes over time, and thought of those individuals whose profession it is to do those mosquito activities as an end user in this project, along with the increased awareness that our marshes are faced with threats from sea level rise, and that a number of different marsh enhancements and restoration projects are also all kind of converging and coalescing along one marsh platform throughout the coast of New Jersey, and thinking about how those interactions work together and what it means for the future of our salt marsh, especially as we think about the continuing and changing threats of climate change over time, really form the basis for this project. It's my pleasure to be able to talk about some of the people who are part of this project. Our end users spanned a few different types of individuals. On the left of your screen over here, you'll see the diverse array of mosquito control professionals from up and down the various areas of the coast that we were able to work with on this project. So we had a great representative from them. And then we also worked with a variety of land managers and restoration professionals who kind of serve that role of how our restoration individuals who are looking to enhance or to bring back some of the functions of the marsh approaching these topics, along with mosquito control and climate change impacts. We also had a really awesome project team from the science side, and you're going to hear from a number of these individuals today. I know we're also joined today, in addition to the members you're going to hear about, from one of our technical staff members, Rachel Saccatelli, and you'll hear from directly Dr. Rick Lathrop, Dr. Dina Fonseca, and then my colleague Kaitlin at the reserve. And the last thing I'm going to do is just give you an overview of kind of the main topics we're going to cover today. Rick is going to talk to you about the high resolution mapping and modeling that has gone into understanding how our marshes are changing at the face of sea level rise. Then Dina is going to talk to you about eDNA and the enhanced monitoring of the mosquito populations that took place as part of this project. And then Kaitlin will bring us home with talking about the really awesome education and outreach products that were developed as part of this project. I just want to say one more thing before I turn it over to Rick, and that is that I've been part of many science collaborative projects over time. And I would say this is one that really embodied the idea of co-production of knowledge. I think the science team learned as much from the end users and from the Mosquito Commission staff members as they learned from us on the research end. And it really was a true collaborative project where their knowledge on the ground really helped shape the face of the research that we did in this project. And with that, I will turn it over to Rick.
All right, thank you, Lisa. So Lisa went through our conceptual model that we built collaboratively with the mosquito control agencies. But there are four major mosquitoes that, four and then a fifth, but solicitans, Aedes cantator, Aedes tenorhynchus, and Culex salinarius are the four major mosquitoes that breed in our marshes in New Jersey. And so the overarching question was, how could it change? How will it change in the marsh landscape that we're seeing taking place, but we expect to accelerate with accelerating sea level rise, affect mosquito population ecology? So one of the major, initial focus was thinking about that marsh landscape and how information about present day, but also in terms of future. So one of the habitats that the mosquitoes key in on are what we call high marsh. Marshes dominated by Spartina patens and Disticula spicata, as compared to your cordgrass marsh, typical, in terms of alterniflora grasses. And so where we had good information about the extent of our coastal marshes, we had less detailed information about the type of marsh, and especially in terms of the Spartina patens and the spicata. So part of was to create, update our mapping on marshes, and then key in on those particular types. So if you look across the marsh landscape, you can have small patches over here on the right, where of the cowlick formation of the patens and mixed in with cordgrass. And again, you can see other patches of patens. If you look at this high resolution aerial photographic imagery of the marsh system, you can see the ditches, but this light colored marsh is patens marsh as compared to the cordgrass dominated marsh. So using the USDA National Agricultural Inventory Program, the NAIP data imagery, classified out, pulled out the patens, the high marsh, to give us better information about the distribution of this marsh across the state. So that was one component in terms of what's there now, okay? And then thinking about how might we expect this to change into the future. So for that particular component, we worked with a variety of different data sets. So one based on SLAM modeling, sea level affecting marshes model. So some work done by the NOAA. And so this was projected out at 1, 2, and 3 feet of sea level rise out to 2050. That modeling is to provide information about the salt marsh platform in terms of areas that might convert to either mudflat or open water over that time period and under those scenarios. And then we incorporated additional information about the actual shoreline erosion rates based on a comparison with historical photography. And then did some more detailed spatial modeling of that upland forest edge and those areas that aren't marsh now, that are coastal forests, that are undergoing dieback and could be marsh in the future. Again, to provide mosquito control professionals, where is the marsh changing, and areas that might not be on their radar right now in terms of part of their monitoring might become marsh and problematic in the future. So one of the areas that we were seeing changing quite a bit and becoming new marsh and new mosquito breeding habitats is this area along the upland edge. So the basically coastal marsh. And so Rachel Saccatelli, who was as part of this, did her master's thesis looking at this. And so what I have here is a series of different locations that she did more intensive work on. And so, and the yellow line shows the boundary as of 1940, and then the red line shows the boundary as of 2015. And so these are basically areas where the forest was forest or swamp, and has died back and is now salt marsh. So just pretty incredible changes that we're seeing on our coast in a number of areas. Some places, not so much, but other places, it's been pretty dramatic. And so this is just looking at these particular sites. In some cases, the most dramatic, 9 meters per year in terms of this movement of new marsh being formed. And in these new marshes, then the question was, well, what is happening there? So this is just one case study of an area that we did further work on. So this is looking at an area. We got marsh here, wetland forest here. This was inundated by Hurricane Sandy in 2012. And so I just want to key on this area in the red. And so if we zoom in, we have mixed cordgrass, patens, phragmites, dying forests, and then cedar swamp back here. This is Pre-Sandy, you can see this area. Post-Sandy, basically this was storm surge. Cedar is very susceptible to the saline intrusion and was scorched. You can see it all turned orange and then died, okay? And so going there today in 2020, you know, the cedar has died and it's being invaded by Phragmites marsh. And actual sampling of the marsh pools in this area shows the presence of salt marsh breeding mosquitoes. So this is just an example of how our coastal systems are changing in areas that may not have been important in terms of at least salt marsh mosquito breeding, could have been other species, are now changing, and we expect to continue to change into the future. So at this point, I'm going to hand it over to Dina Fonseca, and she's going to delve more into how we go about monitoring mosquitoes.
Hi, hello. So I'm bringing back this picture that Rich showed, and thank you, Rich, because I do the mosquitoes. I mean, it's fantastic that Rick is being able, and his team being able to show these dramatic changes in the sea level and in how the tide reaches the new upper marsh. But what does that really mean for the mosquitoes? Why should we be thinking about salt marsh mosquitoes all the way up in the upper marsh? Well, these mosquitoes, and we actually did focus on 4 of them: the eastern salt marsh mosquito, the unbanded salt marsh mosquito, the black salt marsh mosquito, and finally the brown salt marsh mosquito. There's clearly a pattern here. We have tools for Anopheles bradleyi, a vector of human malaria, but have not yet employed them. This is a even more brackish, so more fresh water than some of the other species. So, but why would a mosquito lay eggs, or why would a mosquito be in the upper marsh? Well, because there are no fish there. So these mosquitoes are actually laying eggs in literally places with no water. We tend to think of mosquitoes as having to lay the eggs in water. They have immatures need to develop in water, and these do too. But the eggs are laid in the upper marsh in just sort of moist soil that then get flooded, and that's the key. Need to be flooded by tidal fronts, or by, in some cases, and we're seeing that a lot, with large rain events. So we needed to kind of put the two together, not just assume that if the marsh has moved, these mosquitoes have in fact moved, but needed to demonstrate they were in fact moving. And to do that, and I'm not being able to, oh, there we go. So it just is sort of showing the kind of the height, the differences in height, and how, okay, and how you have sort of upper marsh and then lower marsh, and the areas that are flooded all the time just don't have these mosquitoes because they have fish. But if you have these beautiful Spartina fields, there are actually potholes in in these areas. In fact, you have to be careful when you're walking so you don't get your foot in there, that have water during the high marsh, the high tide, and so there are mosquitoes there. So how does one survey for salt marsh mosquitoes? Well, traditionally, one has surveyed for salt marsh. I'll sort of get one thing in between. So for over 100 years, the control of salt marsh mosquitoes has been the primary focus of New Jersey mosquito control. You need to sort of, this is a bit of a new slide for this talk, but for this audience, I think it's important to remind us all that salt marsh mosquitoes were a very important part of life before the early 1900s. In the early 1900s, a Rutgers professor in 1904 sort of came up with strategies for controlling the larvae of the salt marsh mosquitoes, therefore preventing them to become adults. It's called proactive control, proactive larval control, because the larvae don't bite. If you have to control the adults, you basically sort of failed in your integrated pest management. Earlier in the 1900s, as well as more recently, the control actually included salt marsh modification. So Lisa showed the pictures of the salt marsh, and you see all these ditching. All this ditching was actually associated with shallow ditching, associated with mosquito control, which basically means that the water, when the tide comes in, the water needs to remain in the high marsh for approximately 5 to 7 days for the mosquitoes to have time to go from eggs to adults, and then so eggs and all the way to adults, and then emerge. The water is receding because the tides only come, these are usually only the very high tides, once or twice a month, the spring tides. And so what Professor Smith realized is that if he were to dig these ditches and the water was made to withdraw faster, the larvae would actually dry out. There would just not be enough time for them to emerge as adults. Could argue that was a very insightful, and it was a very insightful approach, but it does involve significant modification and to some extent drying out of the salt marsh. So later, and it's also a lot of work. So later, in the mid 1900s, so in the middle of the 20th century, insecticides, chemical insecticides were heavily used to control salt marsh mosquitoes. Currently, however, across all coastal counties in New Jersey, salt marsh mosquitoes are controlled by biologicals. So these are Bacillus thuringiensis israelensis, BTI. You may have seen it in the shop as mosquito dunks. You can use it in your home. Yeah, this can be also not the mosquito dunks, but a granular formulation can be applied and is applied proactively across the salt marsh in all of the coastal New Jersey counties. But how does one know where to apply? And that's where knowing how to figuring out when are the populations, you need to apply during the larval stage. Needs to be in early stage because a fourth larval instar is already not feeding. So you really need to have a delicate understanding of where the mosquitoes are, what stages they are. And traditionally, field monitoring relies on dip netting of standard standing water, looking for mosquito larvae. So as an example, this is a dip, and the dip with larvae would look like this. So different counties, relatively standardized across New Jersey, but this is occurring across all the coastal United States. They have different levels of emergency. This would be a major emergency. If you have this many larvae of this size, you're seeing some pupae already, you really messed up. So this would be, usually you see a few larvae, and you would then call your helicopter or some other approach and you apply the BTI. Where to sample is actually being passed from superintendent to superintendent over the years. And now that things are changing, there's a bit of, where are these new hotspots? How does one find the new hotspots in these areas that are extensive and quite difficult to survey? In some areas, especially in urban marshlands, you have a lot of Phragmites. And if any of you have had experience of trying to walk through Phragmites and trying to find a pool to go in and do some dipping, and now you understand why the dip has this long stick, you understand this is not an easy thing to do. So our objective was to develop sampling strategies that would increase the efficiency, the effectiveness of the sampling. So not just tell them, okay, go sample here now, but actually try to come up with better strategies that would not involve such difficult walking across the marshland, but also be responsive to change. So during 4 summers of field campaigns, we undertook different approaches to detect and monitor shifts in the distribution of these 4 of the salt marsh mosquitoes. And these sampling sites were selected in coordination with 5 different county mosquito control agencies. Initially, we basically combed the literature, this is uncharted territory, and consulted colleagues and mosquito control professionals. And I should mention that in areas where effective mosquito control programs are being developed in New Jersey, the life of live larvae in salt marsh mosquitoes is brief, because again, they're monitoring them and they're controlling them. So we tried to use a bit of a longer lasting, trying to cue in on something that is longer lasting. So we try to use egg counts. Later, we've shifted to eggshell sampling, which actually has been used in southern salt marshes. But of note, southern salt marshes are mostly free. They're not composed of the kinds of grasses that we have in New Jersey. They're much more flooded all the time. They're not as tidal. And the sampling, I'm not saying it's easy, but it was basically not doable. And that ended up leading us to choose an eDNA approach. And eDNA just stands for environmental DNA, and it basically means that we're trying to detect the presence of DNA that is being shed by basically any organism that is in an area. I'm shedding DNA, skin cells, organisms in mosquitoes. Every time they change from larval to larval stage, they leave exuvia. That's DNA. Every time they poop, they're leaving DNA. So there's DNA basically all over. It's a forensic approach. You use a quantitative PCR, real-time PCR machine, and it's extremely sensitive. But because it is extremely sensitive, you need to be very careful about making sure that you're focusing on detecting the right thing. And we've had to deal with contamination issues, so it's taken quite a bit of optimization. But I'm happy to be able to declare that we have been able to succeed. How does this aquatic eDNA approach works? Well, you basically focus on an area that may or may not have had larvae. And often you don't see any larvae because, as I just mentioned, they're short-lived. You collect the water, you filter the water through a polycarbonate Trek etched membrane. We optimize what kind of membrane would be better by basically as a way to concentrate the DNA. This membrane is just folded, and then we use a very cheap approach for extracting DNA just from the tip of the little just clip. With sterile, flame sterilized scissors, and using flame sterilized forceps, we extract DNA in basically using a hot shot approach, which is simply sodium hydroxide hot, then stabilized with tris HCl. So basically free, and that's a big deal because it allows us to process lots and lots and lots of samples. Once you have the DNA, you then do the qPCR, the quantitative PCR. You can also do next-gen sequencing for a metagenomic approach. We've considered that, but right now we're done optimized qPCR tests. And we've done that for the 4 species that I've mentioned: Aedes sollicitans, Tenerincus, Cantator, and Culex salinaris. If any of you want to talk to me about exactly how we did this, we focus on the ribosomal RNA interspacer 1 and 2. These are well known to be highly conserved within a species, but highly different across species. Difficulties with using this locus, but we were able to overcome those. And as I mentioned, we now have the assays. So what we then realized is that we needed sort of a next step, because being able to sample for water across, because you need to do this across many different locations to have a more quantitative view of the presence and absence of these mosquito species. At this point, we're not using a quantitative approach. We're basically saying sampling at multiple locations and then either detecting it or not detecting it, and then using how often it is detected and where to get a quantitative view of the presence of that particular species. So that's a lot of work. So in collaboration with Greg Williams from the Hudson County Mosquito Control Program that has been using drones to control mosquitoes, we reached out to him as a way to try to develop a way of sampling for water, to be able to test that water with our eDNA probes. And I would like to thank Rick Lathrop for letting me be the person associated with this particular set of slides, because this is cool. And he was the one that was in the field doing all the, because he was also collecting by hand as a proof of concept while this was going on. So in 2020, we sort of targeted this approach using drones, and these were collected as pairs of samples by hand, as I mentioned, Rick, and by drone with Greg Williams on the driving. Brought back to the lab, filtered that day, and then immediately also tested with our qPCR primers assays, which were already optimized. Oh, I'm sorry, I don't know how to go back. So one thing I should have mentioned there, oh, thank you. This was actually something that Greg optimized. It's basically a 50 mil tube, and he created this was using a 3D printer. He created this system so that when the tube sort of falls on the surface of the water, it sinks. It starts to fill up with water and sinks, and then the drone picks it up, and this little lid closes the tube, and then it's brought out to somebody near the drone to then, with bleaching and sort of really careful about contamination, putting the samples aside, then bleaching, cleaning everything, putting a new 50 ml collection vial on, and the drone goes and collects another set of samples. So I'm now showing you some videos of kind of what it looks like. Now it's pretty small, so keep an eye. I can sort of repeat. I'd love to be able to see all of you, but I'm just going to assume I'm going to slow because this is cool stuff. So focus right here. Here's the video, and so the water, the thing was underwater, just being picked up by the drone, and it's then taken to the person, the handler on the street. And here again, it's fast. So focus. So this is now, here we go. There's the water sample just nicely being picked up through a really dense stand of Phragmites that would have made it almost impossible for a person to get in there. So there are more videos that you can see here on YouTube. And we're really delighted to have been able to set up this collaboration with Hudson County. Again, it's really one of the great things about being in New Jersey is being able to collaborate with a lot of programs. I also am doing extension in general in collaboration with mosquito control programs, and here we go. We have the ability of collaborating and using drones to help them also come up with strategies that will facilitate their lives. Okay, so I am now going to introduce Kaitlin. That is, or I'm going to pass it on to Kaitlin, is going to be talking about the educational programs that she's developed around this project. Thank you.
All right, so hi everybody. Once again, my name is Kaitlin Gannon, and I'm from the JC NERRS, and I help develop the education and outreach products associated with this project. I'm trying to move on to the next slide. There we go. All right, thank you. So do I have, there we go. So one of the things that our end users expressed was an update in the educational and outreach materials. And one of the first things that we wanted to tackle was outreach materials, especially. So the first thing that we did was create an infographic. And the infographic will be a versatile tool. It can be for digital and in-person outreach. So you can see our infographic are 3 panels. The first one discusses the life cycle of the mosquito, how to protect your family, health and safety. The second panel is what to do if you're bitten, ways of preventing mosquitoes from around your home with dumping containers, especially if you're living in an urban area. As well as the different species of mosquitoes, especially if you're living in different habitats by the shore, in the woods, what kind of species you might find there and how to protect yourself against those species. The third panel is about mosquitoes and climate change and the different factors that impact their population size, adult survival rates, and disease transmission. So each one of those factors include intense weather, sea level rise, habitat range changes, and invasive species. So the infographic was, again, we want to make it a versatile tool, both digital and in person. So the infographic itself can be on a website and downloaded or in an email format. But we wanted to do a little bit more than just that. So each one of the graphics that you see in the infographic is actually also can be individually uploaded by the end user. There are about 27 graphics in total. So you can look on the top left corner here. We have some of those images of the mosquitoes. So if one of our end user wants to make a social media post or print out flyers, they can use those individual graphics to help them achieve that. And then as far as in-person outreach, we're thinking festivals, fairs, anything where you're interacting with people one-on-one. We wanted to make the infographic become a trifold display that they can place on their table. So you can see the infographic is in a pop-up trifold. And also we wanted to have it in a brochure format. So you actually have an example of one right here where it folds out and it talks all about the things that are in the graphic, but just in a brochure form. Also, part of the education and outreach tasks for this was to create new education modules for teachers. One of the other things that we discussed with end users was updating some educational lessons. So if any teachers approach them on lessons and activities they can use in the classroom, we wanted to provide them with some new materials. We decided to create 2 activities that were focused on the eDNA analysis, which Dina just described to you, and the DNA techniques that they use to look for those species-specific mosquito larvae. So along with the 2 activities, there's also background information, critical thinking questions, as well as background on the project itself and the science collaborative effort. And we also wanted to make sure that these lessons correlated to next generation science standards. Also, all of these lessons are for grades 6 and up. You can see there's a clip of the first activity, which is sort of an introduction into DNA and DNA extraction. So it's called What Does DNA Look Like? And it's a very simple experiment using household materials and a strawberry or some other soft fruit. And you break up the strawberry and extract the DNA from the strawberry. And we thought this was important, especially as a lesson to start with. It kind of mimics what eDNA analysis is like in isolating that DNA before you analyze it. And then our second education activity that we created is called Muddy Genes. And this one is really mimics the eDNA sampling methods that were done in the project. And we wanted to have the students sample different zones of the salt marsh for different kinds of eDNA. So just to describe this real quick, the activity involves a couple of buckets, some beads, and a pipe cleaner. And each one of these buckets represents a different zone of the salt marsh. Bucket A is the upland marsh or high marsh, B is low marsh, and C is along the muddy creek edge. And within each of these buckets are eDNA sequences or little pieces of eDNA. And the eDNA is represented by beads on a pipe cleaner. It kind of mimics like a little eDNA sequence there. You can see the example in the bottom right corner. So there are 12 different kinds in each one of these buckets, and students will grab in the bucket, pull out a couple at a time, and decode them. So this student right here pulled one for snowy egret, and they can tell because they have this eDNA probe worksheet that helps them match which one correlates to which critter. So they look through each one of these buckets and they record all of their findings of what critters are found in which marsh zone and how much DNA they found from each critter in each marsh zone. So they record all of that, they answer questions. And one thing that's particularly important is the salt marsh mosquito, Aedes sollicitans. Where was this critter found? Which zone? Was it A, B, C? Maybe it was multiple. And how does this information, how is it relevant to marsh mosquito management? So that's an important question at the end of that activity, and also very much relates to the project. So if you have any questions about this, I'll be happy to take questions after the presentation.
So thank you, everyone. I think our team's going to reassemble, and I believe Nick was going to field questions.
Thank you, Rick. You are correct. Getting all my stuff ready here. Got a few questions rolling in already. Megan is doing a nice job of putting these into another document for me so I can read them out to you. The first question we have is related to the modeling. So for the SLAM modeling, how many different data elements were run and approximately how long did it take to process? How long did it need to process?
So we were working with SLAM model results produced by NOAA and then integrating those together. So we didn't run the SLAM modeling per se, but used results that they had created, and then worked with 3 different scenarios in terms of 1, 2, and 3 feet using a 4 millimeter per year accretion rate, and to project out in terms of identifying some of those locations of future change. And then looking at the upland areas, we did some of our own spatial modeling in terms of those areas where marshes could be retreat inland or migrate inland, impeded or versus unimpeded, to add to the SLAM work that NOAA had undertaken.
Thanks, Rick. Just as a quick reminder, you can go ahead and feel free to type questions into the question feature in the GoToWebinar console. We have questions lined up already, but don't be shy. We've got some time here to work through questions. So as they come to you.
I forgot to mention the mapping results were integrated on a web platform, and so NJ Flood Mapper to be able to provide access to our collaborators, but really anyone else, and integrate it with other kinds of environmental data.
Yes, good reminder. And I think Megan posted a link to that tool in the chat at some point. But yeah, earlier in the webinar, and we can maybe post that again as well. We have a clarifying question for Dina. Since Phragmites tends to have a flatter marsh surface than Spartina, I would expect there to be fewer or smaller puddles for mosquitoes to breed in. Have you found that to be the case, less eDNA in Phragmites patches?
We have not done it in such a qualitative or quantitative way. And I wouldn't necessarily assume that the size of the pool would be related to the likelihood that a female would lay eggs there. That's one of the things we really would like to understand, and was part of our original wish list, was to try to understand what are the characteristics of the salt marsh soil that makes a female ED sollicitans or cantata or any of the other ones decide, yep, this is a good place to lay eggs. And it's not necessarily the size of the puddle. It's more likely to be associated with how long that water is going to be there. And so we've talked about looking at microbiome, talk to me if you're interested in those kinds of questions. But that's as far as I can tell you right now.
Thanks, Dina. A couple more questions here coming in. Do you see this research having implications for other types of salt marshes, like those in the south that you mentioned are structured differently?
Is that for me still?
Either. Yeah,
I probably should ask that to Rick, because it has to do with how is this. What do you think, Rick? I mean, the salt marsh, the sea level change is really happening more up here, right?
Yeah, it's a coast-wide phenomena in terms of marshes, and so each marsh has its characteristic vegetation types and the like, and are changing somewhat differently. But we're also seeing changes in terms of mosquito populations, or we would expect to see changes in mosquito populations as well. So I think the overall framework would be adaptable to other locations, and these 4 mosquitoes that Dina mentioned are common up and down the eastern seaboard.
Yep.
Thank you. Sorry, I'm bouncing back and forth here. Okay, so was the color of the aerials used to differentiate between alterniflora and patens or spicata zones of marsh? I think that has to do with the flagging, the images earlier in the
Sure. So the NEIP data comes in basically a true color and then a false color. In the newer versions include a near infrared band as well. So I was working with the 4-color true color plus the near infrared band to be able to make that. And it's leaf on. Basically, the imagery is collected. It's about a meter scale in terms of ground resolution cell, and it's collected leaf on, usually August, early September.
Thank you. Okay, so I have another question that's kind of more about the impact of the project, and I'm hoping that maybe Kaitlin can speak a little bit to this. What do you think the biggest impacts and outreach for this project have been? Have you seen anything in particular that's worth noting?
Well, these products are fairly new. I'm not sure if the end users have had a chance to really get them out into the public quite yet. But hoping that it can change perspective of, mosquitoes in and around your home, depending on where you live. There are mosquitoes, and how to deal with that, and how to deal with the changes, and think about these changes moving forward, I think.
Maybe Dina could.
If I may chime in, I mean, COVID, right? So we haven't been able to go to anybody and distribute anything. So that's been a problem. But what I really like and about these brochures is that they go above and beyond the classical, just get rid of the containers and, tip and toss. They try to sort of inform the public about that there's 63 species of mosquitoes in New Jersey alone. And that different mosquito species have going to come out of different kinds of environments, and you need to have different approaches to how to deal with them. And also another very important thing is inform them in terms of broader implications that there are 21 county mosquito control programs across New Jersey, one in each county, that often people are not aware of. And they should contact their local mosquito control programs before, go a bit more radical on their approach to mosquitoes. So it's a nice brochure because it really creates a lot of, is providing a lot of information. I'm curious to know how the teachers, if anybody in the audience would like to chime in, this is a two-way street here. I'd be curious to know how this could be integrated into a science curriculum. I think Kaitlin does a really good job of making it interesting and approachable.
Agreed.
And
Lisa, yeah.
Lisa, go ahead.
Oh, no, fine. I was just going to add that to underscore what Kaitlin said prior about the infographics being developed in a very versatile way, so that the goal is not just that we pass the material on to the mosquito commissions, but they now have a repository of 27 different really pretty nice looking infographics that if they're going to tweet or do a Facebook post or Instagram and connect back with their own county visitors or residents, that they're also sort of in an exponential way getting the message out as well. And I believe we're making those graphics available to anyone who wants to use them. So if other people are interested in mosquito graphics, I believe they'll be part of the follow-up materials that are going to be included on the Neuroscience Collaborative website.
And then Dina, I don't know if you could comment a little bit about in terms of thinking about eDNA and how the mosquito agencies could employ this kind of new tool. And so working with our collaborators, been a lot of thinking about what could it be used for, how could it fit into their normal operational.
Yeah, that's I mean, it's tricky because it involves a molecular laboratory, which many mosquito control programs don't have, but we have sort of similar, the testing in mosquitoes for West Nile virus and many other arbovirus and pathogens is done by the Department of Health. So it's a central location. The mosquito control programs do the surveillance. They send the mosquitoes as pools. They're tested, and then they, within a couple of days, 3 days, well, depending on COVID, they get back, and then mosquito control programs can make decisions about how to act or not act as a response. So This is something we as you know, it has taken. This was in many ways a little surprise to me because I knew that Aedes sollicitans in particular was such an important mosquito. I didn't realize how little there was available about their genetics, and so we really had to kind of start from scratch. So it took longer than we were hoping it would take. And I'm hoping now, and I've had good conversations with members of mosquito control programs, State Mosquito Control Commission, that are intrigued about the possibility of starting to do broader scale, especially trying to find these new locations that, you know, Rick has already identified as, or Rick's team identified as where mosquitoes are coming from, mosquitoes of importance are coming from. So as we make that more evident, they've been very flexible about trying to discuss ways in which they could integrate this into their, you know, day-to-day. They don't like walking around in the summer across the salt marsh looking for mosquitoes. Maybe a little bit, but not that much. Hope that answered your question.
Any other comments? We have another question. So just before we move on, I'd rather to give everyone else a chance to comment if they had anything else to say. No? Okay. So another question. Does an increase in mosquito populations co-occur with other climate change events like harmful algal blooms or Vibrio increases?
I never thought about that. Well, I mean, I think they're two independent things. I mean, they're all dependent on temperature and probably I don't know much about Vibrio. So we know that mosquitoes are temperature and water dependent. Humid environments and warm environments lead to, in general, and in particular, they lead to the large populations of nuisance mosquitoes. We do have some snow pool mosquitoes that are coming out, will be coming out in a few weeks, that absolutely hate this entire climate change warming thing. They don't do well in warm temperatures. But we do have, in general, insects, if the conditions are warm, will develop faster. That means they can have more generations. Again, some mosquitoes only have one generation no matter what, but a lot of the ones that are a nuisance, including the salt marsh mosquitoes, have multiple generations. So in general, warmer, wetter climate is going to lead to larger populations of the existing mosquitoes. It also is leading to the expansion north of southern species of mosquitoes. So I mentioned 63 species in New Jersey. Well, there was just a new one found, a more southern mosquito found in New Jersey. So, and this is happening, and we're finding them, you know, New Jersey is this nice rectangle that is really a great location to look at how changes in temperature is affecting the distribution of species. And we're seeing more and more of the species, southern New Jersey moving into northern New Jersey, and then southern species that were not in New Jersey occurring in New Jersey. So if that's connected to algal blooms and Vibrio, it could be coincidence. So it's temperature.
And the aquatic habitats where they're breeding in could be in terms of inundation of saline water or rainwater. So depending on those other factors that are taking place in the estuarine system, related to how much rainfall events are like. I'm just not familiar with.
But what's interesting? I mean, one thing that has been salt marsh mosquitoes populations have actually been decreasing across New Jersey. And if anybody in the audience there, they can't talk, can they? Well, that's nice. But the fact is that they've been declining, and part of it could be, so some of it could be, and it is, good mosquito control, but also as you get more and more flooded environments, and you're starting to, Rick mentioned this, it's starting to hit areas where there is no possibility of a higher marsh to occur because of construction, because of roads, because of other things. You start getting so much flooded environment, you may actually have less of these salt marsh mosquito species, at least in normal places. But in other areas, they may be, that's what seems to be happening, is you're starting to have an increase. And so it's this shift that usually is complicated because people have to adapt. And it's like an invasive mosquito. The Asian tiger mosquito changed all our lives when it got to New Jersey. Also, if you're not used to having to deal with salt marsh mosquitoes, you may be going for a rude awakening. And you should support your local mosquito control programs, because they can act, and call them. It'll be good for them to know if there's a new active site that they didn't know about. So collaboration.
I just want to point out also a lot of the things that are being mentioned about these other climate change factors, these other climate change things that are occurring could be a factor for breeding and disease transmission. That's outlined in the infographic on the 3rd page. And a lot of it, whether it might affect salt marsh mosquitoes, an intense weather event, maybe in an urban area, can produce more mosquitoes in another area. So these are all these things that we have to think about, depending on where we are, depending on the mosquito, depending on this or that. And that's outlined also, important part to outline in the infographic.
Very good points. We're just about out of time, so I want to just kind of give us one last question to maybe end on here, and then I'll do a quick just outro of what to expect next month and the next couple days. So one last question that I will give you all like a minute or so to answer is, where do you see this work headed next? Just you can maybe each speak to a different piece of where do you think the next steps are for this?
Rick, you want to take it?
Okay. We're going to continue to look at refining models on how the marshes may change and better understanding those aspects. So from the remote sensing side, and then as we develop more information on where the mosquitoes are breeding, we could get a better handle on predicting those future hotspots. We've made a start, but definitely more to be done there. So I'll hand it to Dina.
Yeah, I mean, we would like to understand a little bit better the longevity of the DNA, and so we've done some experiments in the laboratory. We already kind of knew that salt water actually it preserves DNA, but temperature destroys DNA. It's just bacterial growth, which is the primary thing that destroys DNA. So we would like to follow up, and we're looking for ways of continuing to collaborate with the counties to do some experiments and also to look at longevity, because it will give us a better understanding of when you're seeing a signal, is that an active population right now, which is what that's what our preliminary data are telling us, or is this just a remnant of an old population that was already controlled and you shouldn't be worried about? So those are 2 important things we need to separate. To be fair, when I first started this, I thought there's going to be DNA of these salt marsh mosquitoes everywhere. There's not going to be any signal whatsoever. And instead, we found the opposite. So it does seem that the DNA is degrading really quickly. And so when we see a positive, it seems to be very tightly connected to an ongoing infestation, which often is felt by the people on the ground trying to get the samples, because these mosquitoes are very happy to bite people. Although they're bird biters, they're also, if you go into their territory, they feel it was your choice. So you're going to get a blood meal from you. So there's definitely, this is a great start. And again, the collaboration with Hudson County and the use of drones, I think it's a big a big positive for the other counties to try and to think about using something like drone sampling, and could allow us to have a much more extensive sampling and better understanding of the mosquito salt marsh mosquito populations.
Lisa?
I think for me, the thing that I find probably the most complicated and also the most intriguing is the interrelationship between the desire to want to restore and enhance the marsh, trying to combat climate change, and also the Mosquito Control Commission members having to do their important job to maintain and protect public health, and like that whole mix of multiple things all happening in the same space. I think there's a lot more there to unpack, and it's going to constantly be changing with climate. So I think there's like never ending science collaborative proposals there.
Kaitlin, you get the last word.
Sure. So, I mean, next steps for me is just to, get the products out there, especially working with teachers. I'd like to hear their feedback on these activities. And I know for sure teachers are going to be interested in it because it does bring a lot of real world application to it, which teachers are looking for when they are doing activities in their classroom. I've sort of held off on presenting the materials to teachers just because they are quite overwhelmed right now. But I'm looking forward to hearing their feedback, and maybe we can do some extension activities sort of in the eDNA realm, which will be really exciting.
Awesome. Well, thank you all. I know we're out of time here. I just wanted to make sure that everybody had a chance to weigh in on that last question. So thank you all for joining us for today's webinar. Be on the lookout for the recording and the slide deck within the next couple of days. We posted to the Science Collaborative's website. And don't forget to join us next month for our next webinar, which will be put on the calendar shortly. We're still ironing out some last minute details, but you'll receive an announcement about that if you are on our mailing list. And if you'd like to join our mailing list, you can do that at the Science Collaborative's website as well. And I believe those announcements, that link is also in our follow-up announcements as well. So with that, thank you all for joining us, and a big thank you to our speakers for taking the time to put together this excellent presentation and answer our questions for us. And everyone have a great day and stay safe.
All right. Thanks for
Bye,
coming.
thank you.